more. It gives anatomists something to do. Okay, so let's talk about muscle tissue. Now I'm going to tell you this right now. For the majority of this chapter, we're going to be talking about skeletal muscle. Okay, the majority of this chapter will be based on skeletal muscle. Okay, we'll talk a little bit about cardiac muscle, and then a little bit on smooth muscle. But not to worry, you folks will talk more about cardiac muscle, right, when you do the heart. Listen to 11. Right, and then you'll talk a little bit more about smooth muscle. Smooth muscle, when I say it's relatively easy, it's basic. It's an involuntary muscle that just moves stuff through your intestines or your blood vessels, okay? So we already know that there's 3 types of muscle, okay? Skeletal, cardiac, and smooth, right? So your muscles make close to half of your weight. That's impressive, okay? Now, if you see, there's 700 skeletal muscles, right? If you add up all the terms that you have to know for this class, you won't even come to a quarter of that. Thank goodness, right? There's a lot, though. Okay, but what it means is what that 700 skeletal muscles, it's including your right biceps brachialis and your left biceps brachialis. That's what they're talking about, okay? So keep in mind, that's a lot. So first things first, when you think of muscles, you should be thinking of movement, right? That's what it does, right? It's going to create movement in your body, right? So talking about skeletal muscle, right, we already talked about the muscles of the head and the face, right? So those muscles help to move your head, help to make facial expressions, they allow you to actually chew, all right? I'm using mine right now as I'm speaking to you because I'm using the muscles of my larynx, my voice box. And then as I take a drink out of my water here, I'm using the muscles of my pharynx. Those are skeletal muscles that help me to allow me to swallow, okay? Your diaphragm, that's skeletal muscle. Okay, that's helping you to breathe. At least inspiration, the majority of the breathing process, all right, when you're taking a breath in is the diaphragm. Okay, and then exhalation is the passive process. Everything's kind of springing back into its original position. All right, so movement, big function for skeletal muscle. Posture, right now, everybody's sitting here taking notes. We're upright. Okay, so you're engaging skeletal muscles right now. Erector spinae, quadratus lumborum, core muscles, several of the other back muscles are being engaged right now as you're sitting there listening to me. Okay? Protection and support. This is one of my favorite ones to talk about because that's what killed Houdini. There was a movie on the History Channel a couple of years ago. Adrian Brody played Houdini. Are you familiar with Houdini, the escape artist? His big thing, he was big into, what he really enjoyed was, he was the guy that wanted to tell you, if you did a magic trick for him, he wanted to be the person that told you, I know how you did that. That was his thing. He was big into it as an escape artist. Well, anyways, another thing that he was into was letting people punch him in the stomach. Yeah, that was his thing. He bragged about how strong his core muscles were. I got strong, you call him abs, but he goes, I got strong stomach muscles. So he'd let people punch him in the stomach, and he was quite strong there. Well, he asked one guy, dared one guy to do it, and this guy did, punched him. He didn't contract his muscles in time, caught him off guard, and ended up rupturing his spleen. But then he let the guy punch him again because he wasn't ready. So unfortunately, that's what killed Houdini. His spleen ruptured and he died. All right. But the point is, I bring that up is because these muscles help to offer protection a lot for those internal organs there. Okay. Also, elimination of materials here, your sphincter muscles, right? When you're learning, all right, how to go potty, all right, when mommy and daddy are toilet training you, you're learning how to use your external urethral sphincter muscle and your external anal sphincter muscle because that allows you voluntary control over urination and defecation, right? And then heat production, shivering, we talked about that first chapter. Remember, we were talking about thermal regulation, what happens when you're in a cold environment, your skeletal muscles start to shiver. When they shiver, right, they're generating heat from that friction of the contraction and relaxation process there, right? So heat production is another process. So the 5 major functions of skeletal muscles are body movement, maintenance of posture, protection and support of our internal organs, right? Storage and movement of material, and heat production, right? Those are going to be your 5 major functions here. Okay, we talked about the functions. Now we're going to talk about the characteristics, all right, of skeletal muscle. Okay, so and again, we're talking about the characteristics of the nervous tissue, right? And so there's a lot of similarities. First one we saw is shared by both nervous tissue and skeletal muscle tissue. Excitability. Okay, so a stimulus, okay, some sort of stimulus is going to excite our muscle tissue to engage in one of its functions. Okay, so usually the stimulus will be an action potential. Okay, so that's what we're talking about here, changing of the electrical membrane potential. So guess what? Our skeletal muscles have a membrane potential like our nervous tissue did, our neurons. Okay, so we're going to talk about that. Conductivity, well, guess what? Our neuron had the same characteristic listed, right? So it involves, right, that transmission or that movement of that electrical change, that charge that's going to go down the cell membrane. Okay, we saw the movement of a graded potential in the neuron, an action potential in the axon. Okay, well, we're going to see very similar type of concept occurring in our muscle tissue. Okay, so a new one here that we didn't see with the neuron is contractility, muscle contraction. And we're going to learn what actually causes that muscle contraction, which this just reminded me. I'm going to finish this slide up, and then I've got to show you guys a quick video. Okay, I want to show you this video. Okay, so we're going to talk about these filaments. They call it the sliding filament theory. Okay, in which these two proteins, right, actin and myosin, we talked about them. They're myofilaments. We talked about myofilaments in chapter 4. They slide past each other. Okay, and that's what causes, right, that contraction. What causes that movement. Okay, extensibility. That's very important. That gives our muscles the stretchiness, right? So they can be stretched, they can contract. What about the opposite of contraction? Stretching, okay? So when we see contraction, the cell gets shorter. With extensibility, the cell gets longer. And finally, elasticity, right? We eventually want that muscle to rebound to its original shape, like your athletic shorts or your underwear that has an elastic band there. You want it to recoil back to its original shape. That's what we're talking about with muscle characteristics. That it can, after it's been stretched, it can rebound to its original shape, or when it contracts, that it can rebound to its original shape. So that's its elasticity. All right, let me find that. YouTube. Give me a second at home, guys. Oh, it's not on my page. You guys want to watch the Black Widow trailer? Just kidding. All right, short video, which I like. I like short videos. This is the one. All right. You do use muscles, but let me share this with the rest of the class. All right, let me pull this up for y'all at home. Okay, so you put your pens down for a moment. For 4.5 minutes, we're going to listen to this video here. Okay, and by the end of this chapter, you will understand every single thing in this video. It's going to blow your minds. All right, so if this video overwhelms you, that's normal. Okay, let's do it.
Muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction.
So we already know this. Okay, this is what I've been talking about. We just learned this in chapter 12. All right, that's talking about the action potential and then what happens at the synaptic knob. Okay, so we already understand this part.
Is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
What do you think? That is a lot. You're going to learn all that, and then some. Right? So I promise you. Right? So you're going to understand that in much more detail. So I will go over this slide again in the next class, and we'll kind of go from there. But just keep that in mind. We're going to learn how all that works. Okay. I know it can be daunting. Trust me. All right. We'll get rid of that. Stop sharing that, and let's go back into our notes. Okay. Again, we'll go over that in more detail, and let's start off by talking about muscle tissue. Okay. So we're going to talk about the actual anatomy of the muscle tissue. Okay. Oh yeah. Buckle up, because here we go. All right. So like I said when we were in chapter 7 talking about how bones are organs, well, guess what? Our skeletal muscle is the organ for this system that we're talking about. Okay. So keep that in mind, right? Skeletal muscle is an organ. So as you know, organs have several different types of tissue involved. Right? Obviously, we're going to have the skeletal muscle tissue, right? But not only that, we're going to include connective tissue, because there's certain layers. What attaches muscles to bones, or muscles to skin, or muscles to other muscles, tendons? Well, that's connective tissue. So we include that here in this chapter. Then, of course, we can't forget our blood vessels and our nerves. Our nerves are going to stimulate muscles, right? And then the blood vessels supply nutrients to the muscles and actually remove wastes. So this is what we're talking about. All these different types of tissues here. All right, so let's start with the most basic. Right, when we talk about a muscle fiber, okay, we will have, and I'm going to go from big to small. I'm going to break it down. Okay, so our muscle is made up of bundles of fascicles. Okay, and what a fascicle is just a grouping of muscle fibers, and a muscle fiber is a muscle cell. Okay, so let's start off here, real small. Okay, here's our muscle fibers right here. Okay, so this is one individual muscle cell. So now what we're going to do is we're going to package up, right, a bunch of muscle cells, and we're going to make ourselves a fascicle. That's what this bigger cylindrical structure is representing. Okay, that is your muscle fascicle. And then when we package up a bunch of muscle fascicles, we'll have our entire muscle. Okay, so much like with the nervous tissue, remember there was 3 coverings when we were talking about the nervous tissue? You had the individual covering. What's the name of the individual covering on one single neuron? Heard it. I think I heard it. Endo. Did you say endoneurium? Did someone say that? Okay, endo, you know. Yeah, endoneurium. Well, guess what? When we're talking, endo is going to be the innermost layer, but neurium is describing it because it's the nerve. Same thing. When we're talking about the covering to muscle, we call it endomysin. Okay? It's the same type of pattern here. So now we're going to take a couple muscle cells. Okay? You have the neuron, which is the individual nervous tissue cell. All right? Now, here we got our muscle fiber, which is our individual muscle cell. Now we're going to package that up into these fascicles. Right? And then we're going to cover that fascicle, right, with perimyosin. Right? And then when we bundle a bunch of those fascicles together for the whole muscle, then we have epimyosin. Right? And we're going to see that here in a second. So let's go back to this slide here just to make sure everybody knows. Right? An individual, you need to write this down. A muscle fiber is the muscle cell. Okay? Another name for that is myofibers. Okay, so one individual muscle cell is a muscle fiber, also known as myofibers. So when we package up those myofibers, or those muscle fibers, into a bundle, we call that a fascicle. Then when we package up all of those muscle fascicles, right, then we have the whole muscle. Okay, good with that? It'll be a little bit confusing, but it's somewhat similar to the nervous tissue, okay? Very similar pattern there. So we have our 3 layers, which we already understand because it's the same type of connective tissue, okay? In the pattern of it is what I should say. I'm going to start with the innermost one, the endomysin. It's made up of a loose connective tissue, that areolar connective tissue, which goes around each individual muscle fiber, each individual cell, okay? And guess what? It shares a very similar role as the endoneurium did. It helps to insulate that muscle cell, okay? Because we're going to be generating an electrical charge that's going to travel along the surface of that muscle cell, okay? And it separates that muscle cell from other muscle cells, okay? But also you'll find that there's blood vessels in this loose connective tissue, okay? Now we've taken a bunch of these muscle fibers and we bundle them together. And what we wrap around the outside of that is the perimyosin, which is a dense irregular connective tissue. Same thing that we saw with the perineurium. It was also a dense irregular connective tissue, right? But around in the perimyosin, as we saw in the perineurium, that's where we find blood vessels and nerves. Similar pattern. Okay, so there's really nothing too new here except for the name. Okay, then finally, what we wrap around the whole muscle is our epimyosin. Again, it's that dense, irregular connective tissue. Okay, same thing, is that epineurium was also a dense, irregular connective tissue. And all of these layers help mainly for protection, protect that muscle. Okay? Questions? Right. Let's jump into then some of the other connective tissue components that we'll see. Obviously, if we're going to talk about muscles, we're going to talk about tendons. Okay? Tendons are definitely involved. Right? And we've learned in lab, all right, we've got the regular tendon. It's like a cord-like structure that's going to attach, all right, muscle to either another muscle or skin or bone, mainly bone. Right? But also we learned this term, aponeurosis. Right? And we remember from that epicranial aponeurosis or that gala aponeurotica that was part of, all right, the epicranius muscle. Remember frontalis muscle and occipitalis. There was that thin sheet of connective tissue that attaches the two. Okay, well, aponeurosis is that flattened sheet of dense irregular connective tissue, which is made up of all three of those layers that we talked about. That's what that 3CT means, three connective tissue, right? So that epimyosin, that perimyosin, and the endomyosin all will fuse together and form this aponeurosis, okay? If there's an aponeurosis present in that muscle. Right? If it's not an aponeurosis, it could be a tendon. Now remember, tendon is made up of dense regular connective tissue. Okay? But it also is formed by those three connective tissue layers that we just talked about on the previous slide. Okay? As dense aponeurosis. Okay, then that's a misprint there. Go with what the book says. All right, so two new terms that we haven't talked about. Fascia. Have you ever heard me talk about fascia? Oh, you have? Okay. Cool. I didn't know if I mentioned this to you before. Well, if you ever do a wet dissection, when you're actually dissecting cat, dog, well, not dog, but human body or whatever, there's this tissue called fascia. And way back when, they used to think it was just junk. They used to throw it out. You know, they dissect it off out of the cadaver and toss it away. Well, there's a lot more research now that they're finding, especially with the deep and superficial fascia, that if you're having musculoskeletal, like say you're having low back pain, okay, and yet we do all this imaging and there's nothing wrong with your back, right? There is a current thought. What do I want to say? I don't want to say thought process, but well, theory, yeah, a current thinking, way of thinking that it's the superficial fascia or the deep fascia. There could be an issue with that somewhere else in the body. You've got one piece of fascia that goes all the way from the top of your head down all the way to the bottom of your feet. It's one single sheet of fascia, right? And they're finding that you might have adhesions in certain areas of this fascia that could be not radiating pain, but causing pain in other areas of your body. So there's still more to come out on that. So it's still a theory. Okay. Anyways, so the deep fascia, right, is what you're going to see when you're dissecting muscles and you're separating the muscles from each other. Right. Perfect example in your arm, since we've already done the upper extremity. Right. If you're going to be dissecting and separating your biceps brachialis away from brachialis, right, the deep fascia helps to separate those two muscles, right? It's dense irregular, okay? So this is what's going to separate your individual muscles, right? The superficial, right, that's a loose connective tissue and adipose, so very similar to the subcutaneous, all right, layer, right? If you remember, we were talking about the skin, you've got your epidermis, you've got your dermis, and then right below that is the subcutaneous layer, or the hypodermis. Usually right below that is when you start to find your skeletal muscle, okay? So that's what we're seeing here. So it's this layer, this superficial fascia, that's going to separate the muscles from the skin, Okay, and so it's going to share a very similar composition to the subcutaneous layer. Remember, the subcutaneous layer is pretty much made of adipose and areolar connective tissue. Okay, well, so is the superficial fascia. A lot of times when you're dissecting in wet dissections, when you're pulling the skin off, you pull the superficial fascia off too. This comes with it. Okay. All right, and then finally, all right, the other types of tissue are going to be our blood vessels and our nerves. Something you really need to understand. Best skeletal muscle is vascularized. It's highly vascularized. Lots of blood vessels are going to your skeletal muscle. It makes sense, right? Because those muscles, it's a big organ. If almost 50% of your body weight is skeletal muscle, right, you need to have a good blood supply. Okay? So it goes without saying. Right? And it's also going to be innervated, you need to know this, by somatic neurons. Somatic neurons are the neurons that you have voluntary control over. Okay? Voluntary. When you see somatic neurons, you should automatically be thinking voluntary control or conscious perception. If it's sensory, it's a somatic sensory neuron, then you have conscious perception of whatever it's sensing. Right? If it's a somatic motor neuron, that means you have voluntary control over whatever it's controlling. Okay? All right. So now we're going to get into a couple new structures here that we're not familiar with. So remember when we talked about the neuron, we talked about the cytoplasm and the axon, we called that axioplasm. Well, we got a special name for it here in a muscle cell. We call that sarcoplasm. Okay? But what's important about that is that's where we're going to find our contractile proteins. We're going to spend more time on that, right? That actin and myosin. So on that video that you saw earlier, okay, and it was showing the actin and the myosin where they were kind of cross-bridging there, right? That is all going on in the sarcoplasm, going on in the cell cytoplasm, okay? So one of the things that we learned back in chapter 5 for labs is that skeletal muscle cells are long and cylindrical, but they were also multinucleated. And the reason why is here. I'm going to come back to this. Okay? So when you're developing, okay, you have these what we call myoblasts, these individual cells, right? And what happens is they start to differentiate and fuse to one another. And so as they're fusing, they keep their nuclei, right? But if you've ever watched like water droplets kind of move together, or mercury, if you've ever seen mercury on a flat top, it all kind of congeals together. That's what happens here, right? These myoblasts will start to fuse together to form one long cell. In case you're ever wondering, why is a muscle cell have many nuclei? Well, it starts off with myoblasts during your development, right? When you're a little fetus and you're developing, okay, this is what's going on. So they all fuse together. Okay? All right, and then this new term here, sarcolemma, right, is what we call the plasma membrane here in our muscle cell. In the neuron, remember we called it the axiolemma. Okay, well, when it was in the axon. Okay, so the sarcolemma is going to be what we refer to as the plasma, well, that's what we call the plasma membrane. Right? So there's a new structure that we didn't really get a chance. You saw it in the video briefly, all right, which we call T-tubules. Right? And a T-tubule is essentially like a well. Okay? Think of it like a watering well. Okay? It's this hole that goes down into the ground. Well, the T-tubule is this big, long tunnel-like hole that goes down into the muscle cell. Right? And it's going to conduct an electrical charge down into that well or into that T-tubule. Okay? So you can see it on the best. Well, video shows a little bit better than this, but all right, I apologize. I'm going to have to subject you folks to some of my horrible drawings. All right. So here's the top of the cell, okay, the plasma membrane here, or the sarcolemma, and then the T tubule is this big long hole, right, that goes down. And they're all over the place, right? They're scattered throughout the muscle cell. And what will happen is the electrical charge will travel along, and then it'll go whoop, down in there, okay? And it's going to stimulate what we call a voltage-gated calcium channel. And we've seen a voltage-gated calcium channel before in the synaptic knob, remember? When the action potential comes down, it opens up the voltage-gated calcium channel, calcium floods into the synaptic knob, and then that causes the synaptic vesicles to release the neurotransmitter, right? Well, that's what we're going to find. We're going to find these voltage-gated calcium channels down in our T tubule here, right? More on that here in a moment, okay? So keep in mind, the sarcolemma has all these T tubules scattered all throughout, okay? And in those T tubules, we'll see these voltage-gated channels here, okay, that are going to allow, all right, the movement of ions, and we'll get into it, specifically calcium, because on that video you saw we need calcium to enter into the cell, right, to help move those proteins off those binding sites of our actin, for our myosin to bind onto the actin. We'll talk more about that, okay? But keep in mind, in the T tubules, that's where you'll find these guys, these voltage-sensitive calcium channels. I think of calcium in a resting muscle like a princess trapped in a tower. Okay, and we have to rescue the princess. We need to get that princess out of the tower. We need to get calcium. It's trapped in what we call the sarcoplasmic reticulum. I'll show you a picture of it in a second. It's trapped in there. That's our tower. Our tower is the sarcoplasmic reticulum. We got to get it out of the sarcoplasmic reticulum. And we got to get it into, right, the sarcoplasm, into the cytoplasm of the cell. That's where the contractile proteins are, or actin and myosin are, okay? So we're going to go through all that, all right? So keep in mind, sarcolemma, plasma membrane for a muscle cell. And there's all these holes, or these invaginations, okay, throughout the sarcolemma in each muscle cell, and those are called T tubules. They go deep into the cell, okay? And in these T tubules, we'll have these different types of voltage-gated ion channels. The one in particular is this voltage-sensitive calcium channel, okay? Because once this channel opens up, then we can start to release calcium. That's ideal. That's what we want. That's our end goal here, okay? So here we're seeing a picture, all right, of our muscle cell. All right, so here's here. All right, first of all, let me show you. Here is the muscle cell, or no, excuse me. All right, not the muscle cell. This, I'm getting ahead of myself. Hold on. All right, this right here, this whole structure is a muscle cell, a myofiber. Okay. Now, inside of the muscle cell, we have, this is where it can get confusing. All right, these little tube-like structures, and those are called myofibrils. Okay, different spelling. Myofiber is the muscle cell, and then that's made up of all these little tube-like structures inside, and those are myofibrils. So what we're doing now is I'm going to zoom in on this myofibril down here at the bottom. Okay? In the myofibril, basically you have this structure here called a sarcomere. That is the functional unit of a muscle cell. That's the part of the muscle cell that contracts. Okay? And what we've done with the myofibril is that we've just stuck sarcomeres end to end to end to end. We just kept adding them on. Okay? That's what we're seeing here. Well, inside our sarcomere are here's another term for you, myofilaments. Those are the contractile proteins, the actin and myosin. Okay? So these little myofilaments, these actin and myosin myofilaments are found within the sarcomeres. Okay? And then we just stack those sarcomeres end to end to end, okay? And that gives us our myofibril. So on the outside of the myofibril, right, you can see here, right, you've got this tube-like network here, okay? See how it's kind of purplish there? That is our sarcoplasmic reticulum. You're familiar with the endoplasmic reticulum in the cell? It's very similar. It's a tube. The endoplasmic reticulum was a tubular network. Right? That's where we saw protein synthesis occurring for the proteins that were going to be exported out of the cell. A couple other things go on in there too, right? Detoxification, yada yada. Right? So here, our sarcoplasmic reticulum is this tube-like network on the outside of the myofibril, right? But it's sandwiched, right? You can see here at the ends of our endoplasmic reticulum, you've got what's called the terminal cisterna. Zoom in even more. Right? The terminal cisterna are these purple tube-like structures, okay? And then in between this terminal cisterna and this other terminal cisterna is our T-tubule. Right? That's what we talked about. That T-tubule is that little tube, and that tube there is connected to the top of, I'm going to get to that. Yep, I'm going to get to what the reticulum does. Think of right now, the reticulum is our tower. That's where our princess is trapped right now. That's where our calcium is trapped. See, our calcium doesn't do us any good in the tubes. Where do we want the calcium to go? Into the sarcoplasm. Okay? Okay, so these tubes are separate from the sarcoplasm. Okay? So that's what we want. We want that calcium that's trapped inside these tubes to leave these tubes and go into the sarcoplasm here, into our sarcomeres, where our myofilaments are waiting. Okay? I'll get to all that in a moment. Okay? So how I tell folks is, if you look at my sleeve, all right, my arm is going to be the myofibril. My sleeve is the sarcoplasmic reticulum. Okay, see how it's on the outside? Not a part of my arm, it's on the outside of my arm. Right? My arm, right, is the myofibril. That's where the cytoplasm is. So I've got to get the protein that's trapped in my calcium that's trapped in my sleeve, I got to get it into my arm. That's where those actin and myosin contractile filaments are. Okay, but they're not activated right now. The only way we can activate it is if we have calcium and we'll need ATP. Okay, so I'll get into more of the details there. That was a long, drawn-out way. I'm telling you, this chapter is not easy. Don't worry about it. I'm going to hold your hands and walk you through it. Okay, so just a quick review. Okay, our myofibril, okay, is right here. And when we bundle up a whole bunch of them together, then we have our muscle fiber, or myofiber. Okay? And then on what we're seeing here is our sarcolemma, this clear-like structure, and that is, right, the plasma membrane. And you can see all these little holes here. And those are the openings to the T-tubules. Okay? Oops. Okay? So these openings to the T-tubules, there's this little tube, right? This tube that goes down, and then you can see how it wraps around, okay, this myofilament here. Not myofilament, it's myofibril. Okay? Sarcoplasmic reticulum? Oh, the endomysium. Is it? Yes, it is. It covers the plasma membrane. Yep. Just like how the endoneurium covered the neuron. Okay, so this is now, oops, here we go. So this picture here is showing you, here's the sarcolemma, the plasma membrane, okay, the outside of the cell here. And then you can see that invagination here, okay, this is the opening to the T-tubule. And then on either side of our T-tubule, you have this structure here. This is called the terminal cisterna. And I'll show you this in a second here. Right, your terminal cisterna, right, has all these little tube-like structures coming off of it. That's what we saw up here. Okay. Here is one terminal cisterna, and then on the other side of the T-tubule is the other terminal cisterna, but it has the sarcoplasmic reticulum, all these little tube-like structures. Right, but on either side of our T-tubule is a terminal cisterna, all right, and that's what we refer to as the triad. That's what this picture is showing you here. Okay, here is the opening to the T-tubule. Here's our T-tubule. Then on either side, you have your terminal cisterna. I'll talk about what those things do. Okay, so just so you kind of understand. So we talked about the myofibrils, all right? Those are going to be those sarcomeres, right, that are stacked end to end to end, right? And they're going to be inside of those myofibrils are going to be our myofilaments, which are going to be surrounded by our sarcoplasmic reticulum. Okay? So on either end of the sarcoplasmic reticulum, you have your terminal cisterna. So what's the purpose of the terminal cisterna? That is where our calcium ions are being stored. You need to know that. It's a reservoir for calcium ions. Because right now, calcium ions in a resting muscle are separate from the sarcoplasm. Okay? It's not allowed to be in the sarcoplasm. Because what's in the sarcoplasm? Myofilament, actin, and myosin, and those are our contractile filaments, or proteins, I should say. Okay? So we've separated them. Okay? So, our main goal is to get that calcium to those contractile proteins there. Okay? So, when a muscle contraction is done, we evacuate all the calcium that was around those contractile proteins. We put it back into the terminal cisterna. So we have these calcium ion pumps that do that. And we have these proteins that sit inside of the terminal cisterna, called calmodulin and calsequestrin. And their job is to store the protein, is to store the calcium. Okay, until another muscle contraction. Okay? But we need something to stimulate the release of that calcium. And that's where this comes into play, our calcium release channels. An action potential will stimulate the opening of these calcium release channels. And when that happens, calcium will leave the sarcoplasmic reticulum, okay, and the terminal cisterna, which is part of the sarcoplasmic reticulum, and it goes into sarcoplasm. Okay? This is what this is going to show you. Okay, so here again, here's our T-tubule. Here's one terminal cisterna. Here's the other one. So our action potential comes, here's the plasma membrane up here. Okay? The action potential comes along the plasma membrane, sarcolemma, and it goes down into the T-tubule, and it finds a voltage-sensitive calcium channel. And you guys already know from our experience with voltage-gated potassium channels, voltage-gated sodium channels, what happens? Once that charge triggers this channel, right, it's going to cause this channel to open up, and calcium gets released from, here's the terminal cisterna here, it's going to leave the terminal cisterna and go into the sarcoplasm. Okay? And it's going to get released into the sarcoplasm throughout the inside of the muscle cell. And it's going to go and look for, all right, the actin and myosin binding proteins. All right, we'll get into that more. At least tell me, I know this is a lot, that you understand this part so far. Okay? That what we're seeing, hold on. I'm trying to get rid of all that. All right? That the action potential comes down into the T tubule. It will trigger the opening of a voltage-sensitive calcium channel, and that causes calcium to leave, right, the sarcoplasmic reticulum and go into sarcoplasm, right? We need the calcium to get into the sarcoplasm. We need our princess to leave the tower, okay? Right now it's trapped in the tower. The only way to get it out of the tower is to open up these voltage-sensitive, right, calcium channels. Okay? Once we do that, we'll be in luck. Okay? So that leads me to this, myofilaments. Okay? Okay, I'll do 2 more slides and we'll take a break. All right, myofilaments. What are our myofilaments? Those are the contractile proteins. Those are gonna make, right, the muscle contract, decrease the length of our muscle. Okay, and we talked about these previously in chapter 4. Okay? There's 2 types. And the first one is the myosin, which is also known as the thick filament here. Okay, and that's the one, and I'll come here and show you a quick picture of it, and then we'll come right back. Here's the picture of the myosin. That was the one with those globby heads. Okay, so your myosin filament, right? We call it the thick because it's much bigger than the actin, but it has these heads. It's these heads that are going to connect to our thin filament. Okay. All right. So each strand has one head and a tail. So I'll do my best to try to draw. Here's the tail, and then here's the head. Okay. And on the head, there's a binding site here for actin. That's what's going to give us our cross bridge. And also on the head, there's another binding site for ATP. Okay, so there's 2 binding sites. There's an actin binding site, and then there's going to be an ATP binding site, and you want to know that. Okay, so that's where our ATP is going to attach onto. All right, and then it's going to phosphorylate or break down into ADP, DP, and our inorganic phosphate. Okay, there's 2 binding sites. You need to know that on the myosin head. One for actin, one for the thin filament, and then one for the ATP. Okay. All right. The other myofilament is the thin filament. I didn't write it on there, but it's actin. A-C-T-I-N. I want you to think of actin as like taking 2 pearl necklaces and twisting them around one another. Right here. Okay, that's what we're seeing here. Okay, these are these globular proteins, and they're just like taking 2 pearl necklaces and twisting them around one another. Okay. So the 2 types of proteins that are involved are F-actin and G-actin. I always feel like when I'm saying F-actin, it's like I'm saying something bad. You know, F-actin. All right. So anyways, all right. So you have these 2 actin strands. There's F and there's G. Okay. It's the G-actin that's going to have the binding site for the myosin. So that myosin, that globular myosin, is going to attach onto G-actin. Okay. But this last point here, bullet point here, right, is very important. We have 2 additional what we call regulatory proteins. One's called tropomyosin and the other one's called troponin. Okay? Both of these proteins, right, are going to regulate actin's ability to bind to myosin. So what does that mean? Well, in our picture here, right, tropomyosin and troponin are blocking. You can't see them. Well, you can in a little bit. There's a binding site. Okay, so the tropomyosin is this rope-like protein, and it'll sit right on top of the myosin binding sites, an arresting muscle. Okay, so nothing can connect to actin. We can't contract the muscle. Well, on top of the tropomyosin, you've got this cup-like protein, and that's troponin. Okay, and it's got this little cup-like structure here. Well, that cup-like structure, that's where calcium binds to. And when calcium comes in, all right, like in that video I showed you, calcium comes in, it binds onto that cup-like structure, then it causes this rope structure, that tropomyosin, to move off of the myosin binding site. It exposes the myosin binding site, so now myosin can bind to actin so we can get a muscle contraction. That's why you need calcium. If you didn't have any calcium in your body, you wouldn't be able to remove those regulatory proteins. That's one way. But if you didn't have calcium in your body, what else is that going to affect? Heart? What'd you say? I couldn't hear you. That's one of them, yeah. Oh, good. You remember that? That's the one that usually people forget. Yes, you won't be able to clot blood. Okay. Remember what happens down in the synaptic knob, right? You have those voltage-gated calcium channels. If there's no calcium, calcium can't come in. The neurotransmitter won't be released. If you have no calcium, you don't get that. And let's not forget chapter 7, people. Bones. Remember we need calcium for that hydroxyapatite, for the inorganic component of our bones. So now you're kind of seeing why we need calcium. So this is calcium's role here in the muscle. Calcium causes that troponin tropomyosin complex to move off of the myosin binding sites. And so when we have the myosin binding site exposed, now myosin can bind onto actin so we can start our contraction. Okay? So keep that in mind. All right, I'm going to stop here. Let's take a break. I know that's a lot.